Hello everyone, in this video tutorial we are going to learn how we can design our guide RNA for CRISPR-Cas experiment. So we are going to use a very popular tool for this named CHOPCHOP. So we just need to go to this tool and we are going to put down our target gene or target protein. You can also write the reference sequence here or ensemble ID, or directly you can put your gene name. Uh, Here you can select the species you are going for. For this tutorial, we are going to keep it for Homo sapiens, and you can use any CRISPR system you are using, CRISPR-Cas9, Nickase, CRISPR 12, or 13. You can also use Talon. So the purpose for your experiment, if it's knockout, knock-in, gene activation or repression, you can also select that. And you just need to click on find the target site. So there we get our target sequences and graphical representation of our gene. So you can just scroll to zoom in. This red line is the intron sequences, and this blue are exons. These are, if you can zoom, these are the target sequences. You'll be able to see it after zooming. So this much zoom in is fine, I guess. The red are the less unique sequences, and the yellow and green ones will be the more unique target sequences. So, for example, you can go with anything, and if we select this target sequence, you can see the genomic location, plus minus strand, the GC content, self-complementarity. So it should always be the lesser the better. These are the mismatch. We can get the off-target data from this mismatch 1, 2, and 3. And you can also get these efficiency. So you can select one of these target sequence and you can get this data, where you can also design primer from this, and you don't need to use different tool if you are getting your gene amplified and you want primer. So you will be getting the options for primer. These purple are the primer position you can see here, and these are the restriction sites. As you can see, this is TFIL1, this is BSRL, and this is HPY999, this is PSPG1, and the many restriction sites. The green one are again the unique restriction sites, and the red ones are less unique. So you can select as per your experiment. Okay, so these are the target sequences. These are the left and right primer coordinates. You can also see we have the melting temperature here, but I would say कि you should always go for NEB TM calculator for more accurate melting temperature. Here we can see there are no off targets, and you can see the other data for our sequence, the frame shift frequency, precision score, and the frame shift uh, frequency on different base pairs, insertion frequency, deletion, and insertion frequency. You can see the data here. So for going for guide RNA, we will go to our previous page, and we are just going to pick one target sequence and we need to copy that. Let's say we are copying this sequence here, and now we need to go on a different tool, say IDT, that's integrated DNA technology. So you can here go with pre-designed guide RNA, designed custom guide RNA, or here first we are going to go with check the guide RNA for CRISPR-Cas9 system. The species we will select, it will be the same from CHOPCHOP and whatever you are going with. And the input format will be FASTA sequence. We can select here our sequence that we have copied. So here we need to exclude the PAM sequence. This is protospacer adjacent motif, which is mostly NGG, and the N stands for any nucleotide sequence. So we need to exclude that before searching, and we just need to check. It will process and collect the data of on-target and off-target sequences, and a one accurate or most appropriate sequence will be given for your experiment for the guide RNA. And okay, I guess we are here. Yes, so these are the search results. This is our sequence for guide RNA. This is the on-target score and off-target score. So we need to see that on-target score should also be high and off-target score also be high. For this sequence, we have appropriately high on and off target score, and we can see this guide RNA is expected to be good. So if that's not, you have other options. You can go with pre-designed gRNA. But if this guide RNA is fine for you, you can directly quickly order this, the experiment, your tube scale and anything. You can select it from here. You can make your guide RNA, you design your guide RNA, you can order your guide RNA. So here we can see the other option, the pre-designed guide RNA. If if you do not get appropriate guide RNA, so you can directly select your species and input format. We will let it be the gene symbol. So our gene was BRCA2. I hope you know BRCA2 is a gene responsible for breast cancer, and technically it is responsible for DNA repair system, and it's a important part of this. And you can search here and it will process. Here we have the BRCA gene. These are the pre-designed ID sequences guide RNA. You can select the recommended sequence, and let's see, you can also see the on target and off target sequence here. You can select and design the set and order it from this tool also. So I hope you learned from this video how can you design your guide RNA for your CRISPR-Cas experiment. So if you like it, please like and share and follow for more video tutorial. Thank you.